Hi, my name is Shirley French, and I'm helping to bring the Cyanobacteria workshop to you on behalf of the Kingston Field Naturalists. A special shout out to the Conservation Chair, Chris Hargraves, for his support, and also to Holly Evans for networking with a number of the lake associations about this presentation. I've worked as a research technician in aquatic ecology at Queen's, and also conducted my own research on a lake system at Cubes. But that system is quite in contrast to the Cataraqui watershed, which can be quite eutrophic, meaning that we can get high nutrient inflow into the system, and in some years we get potentially harmful blue-green algae blooms. My concerns led to conversations with Dr. Yuzheng Wang in the coffee room at Queen's, and as a result, I ended up helping to sample with Alan Tian for his graduate studies project, and also through Alan, I met Eden Hataley, who was conducting her research on BGA blooms in Dog Lake. So over to Alan.
Thanks, Shirley. Hi, my name is Alan, and I'm a second-year master's student in the lab of Dr. Yuzheng Wang and Daniel Lefebvre in the Department of Biology at Queen's University. So in the summer of 2018, I helped Stephen Anderson, a master's student who was working on a bunch of fish ponds in Hardington at the time with his project, which involved looking at blue-green algal blooms using drones. But we wanted to expand to a more natural field site. So in 2019, we moved on to Dog Lake. However, we realized that drone imaging and our genetic approaches weren't enough to look at the entire picture and measure the toxicity of the water, which was the ultimate goal with regards to human health. And so we entered into collaboration with Eden Hataley and the Ore Hill Lab. On to you, Eden.
Thanks, Alan. Hello, my name is Eden Hataley. I'm a second-year master's student in the School of Environmental Studies at Queen's University. I completed my undergrad in environmental chemistry at Queen's in 2018 and subsequently joined the Queen's Experimental Ecology and Ecotoxicology Research Group under the supervision of Dr. Diane Orahel at Queen's and Dr. Xavier Ortez at the Ministry of the Environment, Conservation and Parks. Under their guidance, I've spent the past 2 years studying the interaction between cyanotoxins, which are produced by blue-green algae, and microplastics in aquatic ecosystems.
First, I would like to share with you some of the terms that will be used throughout this presentation. BGAs are blue-green algae blooms. Technically, they are cyanobacteria, not algae. Phytoplankton means all suspended floating algae, which would include cyanobacteria, but many other taxonomic groups. And HABs are harmful algae blooms. Back in October 2019, an article came out in Nature. Basically, the title sums it up nicely: widespread global increase in intense lake phytoplankton blooms since the 80s. So for this study, they looked at 71 large lakes worldwide. These were lakes that they had data available about temperature, precipitation, and agricultural activities, as well as good satellite images going back 30 years. In 68% of these large lakes, they found an increase in intense lake blooms over these last decades. In summary, from Science News, worth pointing out is the reasons for bigger, denser, and faster growing blooms were different in each lake. Here on the left, we see an image of Lake Erie, and as you can see, it's during an algae bloom. This lake was part of the study. Lake Erie in 2011 had a very bad blue-green algae bloom, as you can see from this picture from National Geographic. What I wanted to point out here is that the bloom looks like pea soup. We have a very large scum zone here where things are being pushed and aggregated together, perhaps by the wind. And this bloom is dominated by microcystis species. This fantastic image of the Great Lakes was circulated to the KFN by Chris Hargraves from the Freshwater Futures newsletter. The white is freshly fallen snow on the land, and some clouds are to the north. But if we focus in on Lake Erie to the south here, we can see that there are streaks of blue-green bands of algae growth in December 2019. It is likely not a blue-green algae bloom because most of the BGA species bloom in warm temperatures. The water at this time of year would be a chilly 5 degrees Celsius. However, a number of green algae species do thrive in cool temperatures as long as there are sufficient nutrients. If we take a look at the depth profile of the Great Lakes from Wayne Grady's book, we can see how Lake Erie on the right here is a rather shallow lake in comparison to the other Great Lakes. It's especially shallow on the western end, where they do get problematic HABs. There used to be a huge swamp that was a natural ecosystem filter, but that was transformed to agricultural land over a century ago. On average, the depth is less than 8 meters on the western end of Lake Erie. In general, lake features contributing to a bloom and used in the 2019 global study for their analysis are temperature, precipitation, and nutrients due to agricultural activity. But also of importance is depth and sediment loading into a basin, and also the disturbance of the sediments once they are there in the lake. Of course, wind action plays a role, and also macrophytes, the large plants. Because the city of Toledo in Ohio gets its drinking water from the west end of Lake Erie, they need to know when potentially harmful algae blooms can be a threat. The National Weather Service website provides bloom severity forecasts for the west end of Lake Erie. A significant bloom of 7 to 8 was forecasted for 2019, similar to levels seen in 2017. This potentially harmful bloom prediction is strongly influenced by high rainfall when it coincides with the time when farmers are spreading fertilizer. Interestingly, when we had potentially harmful algae blooms in Cranberry Lake in 2016 and 2018, these were mild bloom intensity years in Lake Erie. So it'll be interesting to hear the results of Alan Tian and Eden Hataley's research for 2019 to compare with Lake Erie. In Dog Lake, as indicated by the record from the Ministry of the Environment, Conservation and Parks, this provincial government agency is called upon to sample Dog Lake when residents are concerned about BGA bloom intensity, especially at Gilmore Beach, which is a public access point for swimming and other activities. I was attempting to see if BGA blooms were worse during some years. I think there is inconclusive information available here, but I have summarized their reports. 2016 was a year with the highest amount of toxins measured over the years. And here we have levels of 4.5 to 5 micrograms per liter. But in 2018, they sampled early in August and had very low toxin levels. Later in the month, they had 7 independent calls of concern about the BGA blooms, but they did not sample again in August of 2018. In 2019, they sampled in June, and this result is worth noting because microcystis, one of the main microcystin toxin producers, wasn't even present in this June sample. Oscillatoria is another BGA that can produce microcystins, and it can produce anatoxin, just as the species group Anabina. Oscillatoria is, quote, a cooler water species than microcystis, cooler meaning less than or equal to 20 degrees Celsius, whereas microcystis thrives in 25 to 30 degrees Celsius water. So these are some pictures that I took of Cranberry Lake in 2016. The lake had a characteristic pea soup appearance, and under the microscope, the beaded strands are easily identified as Anabina. That is the potential harmful algae bloom in 2016, which started to be apparent in the latter part of August. That summer had what I considered very low precipitation. I remember it well because I was trying to grow watermelons and they need a lot of water. In 2018, around the same date, I found the same cyanobacteria dominating the water samples, that is, Anabina. This was what I considered a dry summer as well, based on my vegetable garden and photos. Personal communication with Dog Lake Association resident Aaron Bonin, 2018 was also a bad year for BGA blooms in the more windswept areas of Dog Lake. In 2019, Aaron was saying that they didn't have a bad year for BGA blooms where they are on Dog Lake. Here on the left is a 2019 picture from Milburn Bay at the end of August. And on the right is a picture shared by Steve Lordson from Troy Lake, which is towards Jones Falls. And as you can see, they do have a bad BGA bloom in late September. Now, if we take a look at temperature and precipitation data from the Kingston Airport, I'd like to compare 4 years, 2016 through to 2019. First, if we look at the temperature data, on the top left panel, we have the months along the x-axis and the hour along the y-axis. Shades of orange and red signify air temperature between 24 degrees Celsius and 29 degrees Celsius. I think you can see at a glance that 2016 on the top left is a hotter year than 2017. 2018 on the bottom left is similarly hot as it was in 2016. 2019, the year of Alan and Eden's research, is a warmer summer than 2017, but not quite as warm as 2016 and 2018. Now, if we take a look at the precipitation in these 4 years and focus on the spring, summer, and fall rainfall, the color green basically is the hourly indicator of a rain event. Comparing 2016 to 2017, I would say that there is more rain in the spring and the summer of 2017. 2018 looks quite similar to 2016 to me, and 2019 looks like it had a rainier spring than 2016 and 2018, but the summer was drier than in 2017. Before we take a look at Dog Lake samples from Milburn Bay in 2019, I would like to point out that Lake Erie had their peak in BGA blooms around the end of July 2019. In the upper left, high cyanobacterial densities are in red and yellow. August was still bad, but had improved over the month of September. This data was available from the NOAA website. Now let's turn our attention to Dog Lake. Here we have the canoe transects from Alan's project in Milburn Bay, 2019. The red dots are where I collected extra samples for phytoplankton counts. The station in the upper right is beside Eden's Site 2. So I will share with you what type of blue-green algae we're growing here. Along the x-axis, we have the four main BGA groups commonly found under the microscope: Anabina, Aphanizomenon, Microcystis, and Warrenishinia. We have the number of either filaments or colonies per milliliter along the y-axis. At first glance, we can see that Anabina species are the dominant blue-green algae taxa in August and September samples. However, these groups are difficult to accurately compare for two reasons. Filaments of Anabina here on the left tend to have fewer cells than a colony of Microcystis, seen here on the right. On the other hand, the cells in an Anabina filament can be larger in size than the cells in a Microcystis colony. If we look at August 29th, the orange bar, Anabina has approximately 100,000 filaments per milliliter of lake water. If on average we have 30 cells per filament, that's actually 3 million cells per milliliter. If we compare that to the Microcystis colonies, where you have, for August 29th, 10,000 colonies per milliliter, and have on average 300 cells per colony, that is 3 million cells per milliliter, the same number as for Anabina. Either way, these numbers for August and September are far in excess of the health unit's recommendation to stay out of the water for recreational purposes when you have greater than 100,000 cells per milliliter. I will conclude my section here. 2016 and 2018 were bad years for BGA blooms in Cranberry Lake and Dog Lake. Most BGAs do best in high temperatures, that's well established. I'm also suggesting in low precipitation years for Cranberry Lake and Dog Lake. These are, however, observations and need more science. Milburn Bay with a creek inflow may have BGAs during high rain seasons as well. Blooms were dominated by Anabina species from August to September, in good agreement with phytoplankton specialists who mentioned Anabina as abundant over many years. Microcystis species are in Dog Lake from July to September. And lastly, on June 17th, 2019, MECP reported the presence of oscillatoria rather than microcystis at a time when microcystins and anatoxins were present in samples. So that concludes my section, and now I'd like to move over to Alan Tian and his research.
Hi, Shirley, and thank you for that wonderful introduction to BGAs. And now I'd like to move on to my portion of the project. So my lab's involvement in blue-green algal bloom began in 2011 when we began investigating the ability of snails to bioremediate blue-green algae through eating it. We started collecting samples from around this area in 2014 for in-lab experiments, and Dog Lake had quite a lot of blue-green algae. And so when my portion of the project began in 2018, I chose Dog Lake as one of my potential field sites. So my 2019 field season began that winter, where I was curious where blue-green algae are concentrated during winter dormancy. This is an important problem because if blue-green algae flow from lake to lakes rather than come up from the bottom, then we would need to treat it in a different way. So we sampled once a week, went out into the middle of Dog Lake, and collected 155 water samples using an auger. We then tested these samples for blue-green algae, and we found that quite a significant amount was present in the lake during the winter. My main part of the 2019 field season in the summer began in May, and I conducted 39 drone flights, doing 75 drone surveys and conducted 37 trips for water sampling. I also took weather data and water quality data. Eden's involvement in this project began in July, and she collected water samples to measure the concentration of toxins at 2 of our main sites. She also examined how toxins bind to 4 different common types of plastics, and on the right you can see her with one of her field assistants collecting plastics, along with one of my weather stations. Eden's involvement was invaluable to this project as she added a dimension of human health, which is ultimately the end goal of our project. And here you can see her collecting water samples while I conduct a drone flight. In contrast, Shirley provided assistance for collecting water samples through using a canoe and did microscopy categorization for different types of algae. So she allowed us to basically validate our drone flights to see if what we saw from the air and through drone was similar to what she could get from a canoe in water sample, which is a traditional method of testing. So again, here's Shirley in a canoe with another field assistant in a pretty disgusting pea soup. Our study was conducted at several field sites. On the top left, we have our first field site at Battersea, where water flows from Lowborough Lake down to Dog Lake. And so we used this as a control to see if any cyanobacteria was flowing down from Lowborough. On the top right, we had our 6th field site, which is Gilmore Beach, where we conducted water sampling in the past. On the bottom right is the location of the weather station. And on the bottom left, we have our 4 field sites at Milburn Bay. We had 3 field sites in the interior of the bay where we collected water samples, and another one on the other opposite side, which represented the rest of Dog Lake. So this is the Gilmore Beach field site. This was ideal because it was a recreational beach that also had a swimming camp. This beach was regularly closed throughout the last few years by August due to blue-green algae. And we also historically conducted sampling here, and we were provided with data by the township of South Frontenac. Our second and main field site was Milburn Bay, and this was ideal due to the cooperation of several property owners who we're very thankful for. And this area was also very susceptible to blue-green algae due to how secluded and sheltered and shallow it is. A major portion of my project was conducted using drone imaging. Drones have been used for surveying for many years now, especially in agricultural, construction, and ecological monitoring applications. Drone imaging provides several benefits over traditional sampling and other remote sensing methods. From the ground, it's very difficult to see blue-green algae. And using drones, we can not only see it in an early stage, but also get coverage of blue-green algal blooms. So to do this, we used the DJI Mavic Pro, a fairly low-cost consumer-grade drone. We took roughly 300 images at our Milburn Bay field site and 700 images at our Gilmore Point field site for every trip. We then stitched our photos together using specialized software, and then we aligned the images. Because our drones were low cost, we had to use these manual alignment steps to ensure that we can compare images. In doing so, we were able to obtain fairly high quality results that were able to look at how cyanobacteria move throughout the course of a single day. In this animation, you can see how over the course of 1 hour, we were able to track how cyanobacteria was blown from the bottom left of this image down towards the shore, probably by wind action. We were also able to obtain very high quality images. This is a very small crop of the prior image, also on September 17, and the same animation. And you can see throughout the course of a single hour, the cyanobacteria had shifted quite significantly. Here are some select results from my drone imaging. On June 6th, we can see that aquatic plants have covered some of the top right area of the bay, which is the northeast. And as we go through 2 weeks later, they've grown quite a bit. And by July 1st, they've covered much of the northeast portion of the bay until July 15th when they've reached a peak. Yet at this point, we still don't see any blue-green algae, which is quite interesting because Lake Erie was reporting blooms by this point. On July 29th, 2 weeks later, we start to see some blue-green algae on the top right of the image beside the macrophytes. Another interesting thing to note was our water testing from this time period on the very bottom right in the midst of those aquatic plants didn't notice any blue-green algae. 2 weeks later, blue-green algae has covered much of the bottom of the image on the south. And then this continues for August 20th. And by this time, some of the aquatic plants have died off. And on August 26th, we see a resurgence of blue-green algae on the bottom right of the image, bottom left of the image. And by September 5th, we've seen our largest BGA yet. Which has mostly disappeared by September 13th. But by September 17th, we again see quite a significant amount of blue-green algae on the bottom left of the image. By this time, we see that much of those aquatic plants in the top right in the northeastern part of the image have died off, while on the bottom left and the southwestern parts of the image, blue-green algae is persistent. So let's zoom into the image, and you can see that the resolution is very high. Through drone imaging, we're able to achieve resolutions of roughly 2 centimeters per pixel, which is good enough to examine algal blooms, but doesn't violate privacy. So we've zoomed into a property owner's dock here, and we can see that there's quite a significant amount of blue-green algae at 8:30 a.m. By 9:30, much of it has been blown towards the shore. And by 11:30, much of the blue-green algae is gone. Our current explanation of this is that the blue-green algae can control its own buoyancy and move up and down the water column, and as wind and wave action and sun becomes stronger, it moves down into the water column. So moving on to September 19th, we can see that the blue-green algae has been confined to the bottom left of the image. And then by September 24th, much of it is gone from the lake and the water has cleared up quite significantly. And this trend continues on September 30th, with much of the aquatic plants also being gone. However, as Eden will show you in her part of the presentation, this doesn't mean that toxins are gone from the water. On October 5th, we see a little bit more blue-green algae on the south part of the image. And by October 8th, we notice quite another big bloom, which recedes by October 10th. And then at this time of the year, the water is roughly 10 degrees. And it's quite astounding to see that blue-green algae is still occurring in the south part of the image in that little cove. In summary of the results from our drone surveys, we were able to effectively monitor the aquatic plant coverage and algal blooms of Milburn Bay, a 20 to 30 hectare area in Dog Lake. We obtained 39 images over the course of roughly 16 weeks, which we'll then later extract for blue-green algal signal, as you can see on the right. We found that emergent plant coverage, such as lilies, frogbits, and duckweed, peaked in mid-July. Visible blue-green algal blooms first appeared in late July, and that Milburn Bay blooms reached their peak between the end of August and the start of September. We also found that different areas of Dog Lake experienced large algal blooms at different times of the year. For example, Milburn Bay experienced earlier blooms than Gilmore Point to the north. We also found that algal blooms persisted in October. This was in stark contrast to Lake Erie. Here you can see 4 different satellite photos between July 30th and September 21st. The dark red represents high concentrations of blue-green algae, while blue and green represent lower concentrations. As you can see, large amounts of blue-green algae were observed in really late July and declined from that point. In contrast, we saw algal bloom coverage peak in very late August to early September. In Lake Erie, very little algal blooms remained after September 21st, while in our Dog Lake system, we found that algal blooms persisted even in late October. However, assessing threat to human health requires additional methods. Drone sampling only allows us to see how much cyanobacteria there is on the surface of the water body, not the actual toxin concentration.
Blue-green algal blooms have the potential to negatively impact the economy, the environment, and human, animal, and wildlife health and well-being. Blooms can influence multiple economic sectors that depend on clean water, including water treatment, tourism and recreation, commercial fishing and agriculture, and real estate. Blooms can influence the aquatic environment by outcompeting beneficial phytoplankton or blocking out sunlight. Additionally, blooms consume oxygen as they die and decompose, leaving areas in the water with little or no oxygen, which can result in large fish kills. Lastly, blooms can influence human, animal, and wildlife health and well-being by producing a variety of toxic secondary metabolites called cyanotoxins. Cyanotoxins make up a large and diverse group of compounds that differ in toxicological properties and molecular structure. They are often broadly categorized by their mechanism of toxicity, falling into 4 main groups: liver toxins, nervous system toxins, skin toxins, and irritants. Some types of cyanotoxins have multiple structural variants. For example, more than 250 structural variants of microcystin have been identified in nature. Cyanotoxins exist naturally within the cyanobacterial cell. Here, they are referred to as intracellular toxins. When the cyanobacterial cell dies, the intracellular toxins are released into the environment. Here, they are referred to as extracellular toxins. Although not as common, some species of blue-green algae are capable of releasing cyanotoxins into the water without cell death. Each type of cyanotoxin can be produced by more than one genera of blue-green algae. And likewise, many genera of blue-green algae can produce several types of cyanotoxins. Microcystin can serve as an example. Microcystin is often described as the most widespread type of cyanotoxin and commonly found in Canadian waterways. Microcystin can be produced by multiple genera of blue-green algae, which are each capable of producing other types of cyanotoxins. A blue-green algal bloom does not always mean a cyanotoxin problem exists. The presence of a bloom is not an indication that a bloom is toxic, but rather it is the first visual indication that a risk exists. Why? Not all blue-green algae are capable of producing cyanotoxins. And even when toxin-producing blue-green algae are present, they may not produce cyanotoxins. Laboratory analysis is needed to determine if blue-green algae carry the toxic gene
and if blue-green algae are actually producing toxins.
Thank you, Eden, for that great introduction to cyanobacterial toxins. And now I'd like to move on to the next part of my portion of the project. Environmental DNA, or eDNA, is genetic material that's collected from a variety of environmental samples, such as soil, water, snow, or even air. And this is instead of being directly sampled from an individual organism. As organisms interact with their environment, genetic material is expelled and accumulates in their surroundings. For example, as a fish swims through the water and as it urinates, bits of epithelial cells will be shed off. Examples of sources of environmental DNA include feces, mucus, gametes, shed skin cells, dead animal carcasses, hair, bits of plants that have been eaten by herbivores. And by analyzing this environmental DNA, we can do a great many things. So we can monitor common species, but also to detect and alter and identify other species that could influence conservation. This allows us to monitor biodiversity without actually collecting living organisms. And so this allows us to study organisms that are invasive, very rare, or endangered without actually finding them or handling them, which can be cheaper, require less training, and be very early on. For example, if you have a single invasive organism in your water body, it might be almost impossible to find it manually. But with environmental DNA, we would be able to detect it. And so by accessing this genetic information, we can make critical contributions to understanding of population size, species distribution, and population dynamics, especially for species that are not well documented. For blue-green algae in particular, we can use environmental DNA to quantify the relative amounts of blue-green algae. So our drone photos showed us the distribution of blue-green algal blooms, but didn't actually show us what species they are. Furthermore, even within the same species of blue-green algal bloom, as we can see from microscopy, some have the potential to be toxic while others don't. In our species of interest, microcystis, there's some cyanobacteria that have a strain of DNA that allows it to produce the cyanotoxin microcystin, while others don't. In our study, we sampled water from 6 sites, as previously mentioned, taking 37 trips to collect 218 water samples. Our samples were then filtered to catch the cells, and then we extracted genetic material. From this, we were able to find the concentrations of different types of blue-green algae of interest. Here are some of the findings from the genetic aspect of our study. Microcystis is a very common type of toxin-producing blue-green algae, and so we decided to focus upon it for our study. To determine the total concentration of microcystis, we looked at the 16S gene. This gene codes for a ribosomal protein, which is a protein essential to life. It's very conserved, and it's often used for barcoding or identifying individual species. And so we were able to use it to identify the number of copies of genes and thus number of copies of cyanobacteria in our study system. So as you can see in site 2, which is inside Milburn Bay, we saw results that mostly matched our drone imaging, with a small peak in late June when cyanobacteria first appeared, and then in late September, we have a much greater peak when the bloom coverage was greatest on the lake. In contrast, in site 5, we have a very early peak in early June, followed by another peak in late June, and then finally another peak in late September and early October, similar to site 2. However, total microcystis does not actually represent the toxic quantities of microcystis. Not all blue-green algae can produce cyanobacteria toxins. Not all blue-green algae can produce toxins even when they're of the same species. So for that, we decided to look at the gene that actually produces the toxin. So this gene is called the MCYE gene, and it's one of the 5 genes that codes for proteins that produces the cyanotoxin. We saw trends very similar to the 16S gene, with peaks in late June and at the end of September. So we also looked at the ratio of the MCYE gene to 16S gene as a proxy for the ratio of toxic microcystis to total microcystis. And what we found was toxicity ratio usually hovered around 35% to 30%. This was in contrast to our earlier studies in the summer of 2018 and the winter of 2019, or the winter between 2018 and 2019, where we saw the toxicity ratio hover around 25% in 2018 and was much more stable, but it was also around 30 to 35% in the winter. This indicates that environmental conditions may have a large impact on the ratio of toxic to non-toxic microcystis and blue-green algae in general. So here are our results from previous years. As you can see on the y-axis, the ratio of toxic to non-toxic microcystis was roughly 25% to 28% in 2018, rose to roughly 30% in the winter of 2018 and 2019, and then became much more variable, but also rose to around 35% in the summer of 2019.
The conditions that trigger or inhibit the production of cyanotoxins remain poorly understood. However, several types of cyanotoxins exhibit allelopathic properties. Allelopathy is the phenomenon by which one plant influences another through the release of compounds into the surrounding environment. Cyanotoxins may play different ecological roles. For example, they may play a role in direct resource competition between blue-green algae and other aquatic organisms. People can come into contact with cyanotoxins in a few different ways, including drinking contaminated water, eating contaminated food, and through recreational activities like swimming and boating. If you do come into contact with cyanotoxins, symptoms vary based on how you're exposed, how long you are exposed, and the toxins involved. Short-term exposure can result in a wide range of symptoms in humans, which can occur within minutes to days after exposure. Long-term exposure to low levels of cyanotoxins is less well understood. However, one structural variant of microcystin has been classified by the International Agency for Research on Cancer as possibly carcinogenic to humans after chronic exposure. Health Canada has set the maximum acceptable concentration of total microcystins in drinking water at 1.5 micrograms per liter. This is considered to be protective of the general population. As a precautionary measure, when levels of total microcystins in treated water are detected above 0.4 micrograms per liter, the public is notified. Health Canada has set the maximum acceptable concentration of total microcystins in recreational water at 20 micrograms per liter, or of total blue-green algae in recreational water at 100,000 cells per milliliter. Should either of these guideline values be exceeded, a swimming advisory may be issued by the responsible authority. However, in general, contact with water where a bloom exists or has recently collapsed should be avoided. Currently, in Canada, guidelines are not established for types of cyanotoxins other than microcystins, because health and exposure data on other types of cyanotoxins is limited. This may change as more data becomes available. For example, in the United States, the Environmental Protection Agency has set drinking and recreational water quality guidelines for two types of cyanotoxins, microcystins and cylindrospermopsin. Treatment for cyanotoxins is complex. The treatment needs to account for different types of cyanotoxins and whether they are intracellular or extracellular. There are no residential scale drinking water treatment systems available that are certified for the removal of intracellular and extracellular cyanotoxins in untreated water supplies. If your private water supply is impacted by a blue-green algal bloom, Health Canada recommends changing the location of your water intake pipe or switching to an alternative water supply. Do not boil or treat your water supply with disinfectants. This can increase the extracellular cyanotoxin level by breaking open cyanobacterial cells. We conducted cyanotoxin analysis on surface water samples from a subset of our study sites to determine if cyanotoxin production was occurring in Dog Lake, and if so, which types of cyanotoxins at what concentrations. We collected surface water samples weekly from August till October 2019 from each site. We completed cyanotoxin analysis on surface water samples collected from Site 2 and Site 5 at the Ministry of the Environment, Conservation and Parks Laboratory in Toronto in December 2019. The cyanotoxin analysis method we used can detect two types of cyanotoxins, microcystins and anatoxin-A, and accounts for both intracellular and extracellular toxins. I will share the cyanotoxin analysis results for Site 2 and Site 5. On the vertical axis is surface water concentration of microcystin and anatoxin-A in micrograms per liter. And on the horizontal axis is date, stretching from mid-August till the end of October. Cyanotoxin analysis on surface water samples confirm that cyanotoxins were present at both Site 2 and Site 5 for the duration of our sampling period. We found both types of cyanotoxins that our method was able to detect: microcystins, highlighted in red on the graph, and anatoxin-A, highlighted in blue on the graph. Cyanotoxin concentrations were higher at Site 5 compared to Site 2, and remained within a 2 microgram per liter range for the duration of our sampling period, with the exception of a spike in total microcystins at both sites in early October. So what did we learn from cyanotoxin analysis on surface water samples taken from Dog Lake and Millburn Bay? There is indeed a cyanotoxin problem. If you draw your drinking water supply from the area, I would caution you to use an alternative water supply during blue-green algal bloom season. If you eat fish caught in the area, I would encourage you to familiarize yourself with how to safely prepare and cook a fish caught from waters impacted by a blue-green algal bloom. If you use the area for recreational activities, note that the surface water concentration of total microcystins, highlighted in red on the graph, is well below the maximum acceptable concentration set by Health Canada. What I believe is the most concerning is the presence of anatoxin-A. Although water quality guidelines for anatoxin-A have not been set by Health Canada, every year it is responsible for the lethal intoxication of different species of animals, like dogs and cattle across Canada. Furthermore, the levels of anatoxin-A at Site 5 in September are consistent with what has been previously shown to induce lethal intoxication in dogs and other smaller lakes. If you see a bloom, err on the side of caution and assume toxins are present. Avoid using, drinking, bathing, or swimming in the water, and eating fish from the area. Restrict pet and livestock access to the shoreline and the water. If you come into contact with contaminated water, wash thoroughly with soap and water and seek medical treatment if symptoms begin to appear. If your pet comes into contact with contaminated water, seek veterinary care immediately. Lastly, I will leave you with some municipal and provincial government contact numbers that you may find helpful in the case that you see a blue-green algal bloom.
There are several takeaways from my section of the presentation. First, visible blooms can drastically change within a day, as we saw from the drone photos on September 17th. Secondly, toxins can persist after visible blooms are actually gone, as we saw from the discrepancies between Eden's toxin concentration measurements and my drone photos. Even when the blooms aren't actually visible from the air, toxin concentrations can still be dangerous. Thirdly, results from large lakes such as Lake Erie are not generalizable to smaller lakes, and therefore more research is needed on smaller lakes such as the Rideau system. And finally, drones provide a citizen science-friendly method for monitoring local water bodies, with the cheapest drone being under $400.
We'd like to thank you all for tuning in to our video and for joining in on the collaboration to try to understand and solve the problems of blue-green algal blooms in our local waterways. If you're feeling a little discouraged about the problem, know that by educating yourself and joining in on this collaboration to monitor the problem in your own lake, you're moving in the right direction of helping solve this problem.
Yeah, I have also learned a lot from this project, but I still have questions, and you may also have questions for us about this presentation. You can either contact myself at the email address given, or Alan Tian or Eden Hataley. I was wondering, Alan, if you have any further information about Dog Lake that you could share with us?
Yeah, so Dog Lake, like many of the other lakes in the Rideau system and on the Canadian Shield in general, is very complex. And to better understand cyanobacterial harmful algal blooms and to predict them in the future, we also need to gather other types of data. So for example, Dog Lake has 3 basins. There's a north basin, which is very deep and is directly connected to the Rideau system, and so has high water flow. There's the middle basin, where our Gilmore Point site was at, which also has a fairly deep middle point, and also it has a thermocline. And finally, a southern basin, which is not very deep and doesn't have a thermocline. So the water column is the same throughout. So the southern basin in particular is very susceptible to not only cyanobacterial harmful algal blooms, but also general eutrophication and high amounts of plant coverage due to its stagnancy. It's also very sheltered from the wind and has very little water flow. So the Lake Partners program collects data such as calcium concentration, total phosphorus temperature, and Secchi depth, all of which are very important parameters to understand lakes. For further information on Lake Partners program, please see the links attached to this video.
Yeah, that'll be interesting to check out. It gives an idea of how eutrophic the system is in different regions. And that would go along well with the additional information that we're hoping to get from using fold scopes. These are basically small handheld microscopes, and I'm proposing some sample dates, as you can see from the page here. August 16th, August 30th, and September 13th, plus or minus a few days are fine. That's so we can compare notes along the Rideau waterway. Other parameters of interest would be temperature if possible, but location we can get from Google Maps. The important components for sharing are the photo of the bloom from about 1 meter away, or a lack of a bloom if it's a good year. And secondly, a photo of your water sample under the fold scope. And that can be easily done. Hopefully you'll follow up with watching the fold scope video after this one. So thank you for your interest in learning more about the health of our waterways.